Hello, this is the chapter 24, Amino Acid Biosynthesis. And so we're going to be talking about how the cell comes in and builds up the individual amino acids, the pathways that converge and be able to convert do this. So there's two components to this. The first one is how do we get the nitrogen for the amino acid, and that typically comes from the nitrogen gas that gets reduced down to ammonia, and then the carbon skeletons, which come from glycolysis and pentose phosphate pathway and citric acid cycle. And so the first part of this is actually it's called nitrogen fixation. And this is done primarily by bacteria as well as certain archaea. And they will come in here and reduce down this from nitrogen gas down to ammonia. The problem is that nitrogen gas, right, it makes up something like 78% of our atmosphere. It's incredibly inert. And so being able to break that down, break that triple bond is incredibly energetic. It requires a lot of electrons, a lot of reducing power in order to be able to do that. The enzyme, the nitrogenase complex has two parts, the reductase part and the actual nitrogenase part. So the reductase provides these electrons, provides the reducing power, and the nitrogenase provides the electrons. And actually, it's for every electron that needs to get put in here, it requires 2 ATP. And so the overall net reaction is the nitrogen plus 8 electrons, 16 ATPs, and then out come to ammonia gases. So for that, and so the way this works is that if you have, you've got your nitrogen gas here, as the electrons come in, and when you try to put in, let's say you want to put in an electron, the problem is going to end up being, right, this, if we put it, say, on this nitrogen, if we put in that electron, you've exceeded the octet, okay? And so what ends up happening is these electrons, a pair of electrons has to get pushed over, and so what you end up with is something like this. And then you've got electron here, plus charge. Excuse me, it doesn't have plus charge. We just add an electron, so now it's got a negative charge. This is simplistic, right? And then if you want to add another electron, right, you're going to end up with and as you're adding these, you can add hydrogens are going to be coming in here, but you need to be able to provide electrons in here to be able to break apart this this nitrogen. Basically, you're trying to put in electrons to exceed the octet for the nitrogen, and that's going to push the electrons onto one of the other nitrogens, and then it can come in and pick up hydrogens. So that's the two nitrogens here. These two nitrogens come from here. These hydrogens here comes from these 8 here, because you end up making an equivalent of hydrogen gas, as well as the 6 you need here, and then you burn 16 ATP. So, okay, so the nitrogenase part, or this nitrogenase complex, is incredibly sensitive to O2. With that, because and so, but we just talked about oxidative phosphorylation. We need the oxygen around in order to be able to fuel ATP, to be able to fuel it, be able to fuel this process. So how do you do this? Well, what you do is you end up separating out, compartmentalizing the nitrogenase complex into a separate component, so that and then you transport the O2 to the ATP synthase through a compound very similar to hemoglobin. Okay, so you escort the oxygen into the into the ATP synthase, that process for the electron transport chain, to be able to do that, to supply the ATP, but without having to kill the nitrogenase. Okay. So the electrons come in into an iron sulfur cluster with that, and so that allows you to accept electrons without having to accept hydrogens with that, and so the electrons can very easily hop on to an iron. You go from iron 3 to iron 2, and then you can move on. You get conformational change, which pushes the so once that comes in, you get that change. That allows the electron to move over towards the I should say the ATP hydrolysis causes this conformational change, which moves the electron over, allows the iron to physically move over towards the reductase domain in order to be able to transfer it over. That electron then actually gets put onto another iron sulfur cluster, which then can go onto a molybdenum, which is one of the few times you see molybdenum in nature. And so it's really this the molybdenum that does the chemistry for this. So this part here, this P cluster, this is what accepts the electrons. The electrons then are then used, funneled to the iron molybdenum center to be able to actually do the chemistry. Okay, so once the ammonia is made, okay, we don't want ammonia, flying everywhere within the cell, okay, because it can react with pretty much with any free ketone or free aldehyde that's in there. So it could say react with the glucose that's in the molecule. That's how they actually make the caramel color for the for like Coke and Pepsi soft drinks, caramel color soft drinks. You come in there, and that's actually what makes the caramel coloring. If you're cooking, it's the amine from the from amino acids, along reacting with the sugars to do this Maillard reaction, which gives you that crust in your food. With that, so what we do, like everything else, if we need to compartmentalize something, keep it in there, we can shuttle it. It allows the shuttling of the molecule much more easily. And pretty much what we're going to be doing if we need to shuttle this amine around is we use glutamate or glutamine as nitrogen shuttles. Okay, so alpha ketoglutarate, we form the imine here. It's sometimes called a shift base. And so you lose two of these hydrogens as well as that oxygen. You lose it as water, and then you can come in here. Remember, this is NADPH, right? This is the equivalent to a hydride, and so that simply goes up. Oh. Anyway. So to make the glutamine or glutamate. Now, if we want to add an amine down here, what's going on with my pencil? Oh, give me a second. Gotta put the tip back on. So if we want to put it down here, so this should be up there. This is not. If we want to put an amine down here, this actually is not. One, it's negatively charged, so it's not going to be able to do. It's not going to be able to be electrophile to be able to get attacked by the, so the amine itself. So by that, so what we have to do is we have to make it into a good leaving group of making this phosphoester intermediate. We transfer an ATP over. This is a really good leaving group, and so when the amine comes in and attacks, up, down, kick away, we're then left with the amine. Plus the phosphate leaving group. So glutamine actually, they can have, we can use this amine. We can also use that amine here on glutamate. Okay, so the the processes. So what we're going to be doing is we're going to be building these amino acids from the various components. Okay, and so oxaloacetate can come down here to make aspartate. That can then be used for each one of these with that components, right? So pyruvate can be used to make these guys, um, so on and so on and so forth. Okay, plants and bacteria, they can make all of these, with that, but animals not necessarily. That all of them. With that, and so what you're going to need to do is you're going to need to be able to, you actually need to know this chart with that. And so the other thing is I forgot to mention is that on our planet the sugars are typically in the D configuration, while the amino acids are in the L configuration. There's no necessarily advantage of D versus L. It was just somewhere along the line they had to pick one, and so in the case of amino acids, they picked L, something like that. And so these are actually going to require some stereochemical control for that in order to be able to do this, to be able to make, because otherwise, if not, you're going to get a mixture of D and L, and if you can only make the L, then you're wasting half of your product. Okay? Now, for human amino acid biosynthesis, there's two different types. There's essential, which we actually get from our food, and then non-essential, which are pathways that we still have available in our metabolism. Okay? You do not need to know this list, but you do probably need to know the difference between essential and non-essential. So essential amino acids, those are what you get from your vitamins, right? And so you often hear that, which means we have to intake those. Okay? There's a general rule of thumb is that the more complex the longer the synthesis, the less likely we are to still have that process in there. And so the essential ones, these are the ones in magenta here. The exception would be arginine, and we'll talk about that in a second. But here, if you notice the non-essential ones, these are the ones that we can make. They're all sort of very short, anywhere between 1 and 5 steps. The really difficult ones, these guys down out here, we don't. We just get those from our diet. It's just too energetically. It just costs too much energy. But if you're a little bacteria and you don't have any say on what you can eat, you're going to need all of those, because if you put in, say, a minimal media, where you have glucose, ammonium sulfate, a little bit of phosphate salts, some minimal salts, you're going to have to make all of your every amino acid in order to be able to survive and grow. Okay, so aspartate, glutamate, alanine. Okay, so these are all produced from their respective alpha keto acids. So for aspartic acid, it's oxaloacetate. So if you just come in here and you're transporting, basically swapping in this amine here and putting it in right there. Okay, so this is these are you're literally getting a transfer from glutamate to aspartate. So with that, glutamate comes from alpha-ketoglutarate, right? So with that, and alanine is going to come from pyruvate. Okay? But they all are essentially the same. The way this works is you do need a cofactor. Okay, so with that, and essentially all you're doing is you have this PMP cofactor. It's held onto inside the active site through through active site lysine. Okay, and essentially all you're doing is you're swapping out what nitrogen is being held by the PMP. So from the glutamate, right, it transfers its amine over onto here. So the glutamate from here at this, and then the lysine comes over here and just releases. We can then transfer that onto our amino acid to go here, and it's a pretty slick little process. This base gets removed, and you go here, there, and then when this reverts back, it can come in and take a hydrogen here and set the stereochemistry right there. Okay, and then this can get hydrolyzed away because this is a reversible process. And then what you're left with is this getting hooked back onto the lysine and the release of the amino acid. Okay. Now the positioning of how this comes on, how this hydrogen comes on, determines the stereochemistry of the overall amino acid. Because at this point, everything's flat. Well, from here to here, so there's no stereochemistry there. And it has to come in this orientation. It has to come in from the back side, so like that. So you get a wedge, a dash amino acid, or in order to be able to make the proper amino acid. So here's the amine, here's the carboxylic acid, the hydrogen as the dash, and then your side chain, which is the R. So arginine comes from aspartate, right? So with that, and so again, you're going to be, if you want to add things onto a carboxylic acid, you've got to activate it first, and this is just this here is just going to be a leaving group. This phosphoester, with that's all this is. It's coming in here, and we're leaving, and then the amine can come in here, up, down, kick away. Some of that, so pretty simple for asparagine. Oh, I should mention. So the yeah, it's to say the yeah. So we use glutamate as our nitrogen source. You could also use, and this is one of the few cases where sometimes you actually use ammonia. For proline and arginine, you actually use glutamate, and you get this essentially, again, you're going to activate this ester here and then reduce it down to the aldehyde. Okay, so it's the hydride that comes in and does the attack. Now you've got two choices. You can either add the amine right there, reduce it down, and then to make ornithine, or you can have this amine come in here and cyclize to it right here. So is it an intermolecular reaction where you add amine from the outside, or it's an intramolecular where it cycles down to make the 5-membered ring on the proline? You just reduce this down with another NADPH and you add proline with that. So now ornithine, so this then goes into the urea cycle to make the arginine. This is the one where the urea cycle is still up and running in our body, so that's why this process still exists with that, because it ties into a second cycle. It's sort of an exception to the rule. Okay. So serine comes from 3-phosphoglycerate, right? So with that, which comes from glycolysis. So with that, it also comes from the Calvin cycle in plants. So with that, and so again, if you come in here, you actually oxidize this in here. So 3-phosphoglycerate. So you oxidize here to make your alpha keto acid, and then you can then transfer the amine onto here. You still have the phosphate on there, but that's a simple hydrolysis to make serine. Okay, so the trick is to be able to oxidize this to your alpha keto acid, and then transfer the amine with that onto there. So glycine, okay. So for that, so this does use serine, okay. But what we really need to do is we need to get rid of this. We need to get rid of that CH2OH group from it, and we don't want to just throw it away if we don't have to. And so what we do is we use a molecule of tetrahydrofolate, and that comes in as you get an equivalent of water, right? So the OH combines with one of these hydrogens here, and you end up you end up stripping out the carbon, which we can then use for later. And then now this one, right? So the glycine just has these. Oops, that's really terrible. You've got the two hydrogens here, so that the one that you had before, and then the new hydrogen coming in. You can then use this. So the tetrahydrofolate, right? So if you need to add a one carbon group onto things, then you typically use tetrahydrofolate. Okay, so we can use that to make things like acetylmethionine. Okay, and so if you take methionine, add the ATP to it, you can then use you then create this. If you had ATP, right? So this is acetylmethionine. We can then use that. This is the equivalent of a this right here is basically a giant leaving group. So for that to be able to transfer this onto a one carbon system. So for that, so after this after this goes, right, we can hydrolyze this to make the homocysteine. The homocysteine then, okay, can then accept the one carbon piece from the tetrahydrofolate after it's come in and stripped it away, so we can transfer it onto the homocysteine to make methionine. And so you can actually use so let's see if we can get this right. So you start with serine, right? And then you strip out, that uses to make glycine, but in doing so, you basically strip out a nitrogen, or excuse me, not nitrogen, a methyl group, and then we can use this to make homocysteine to make cysteine. So we very rarely throw anything away, right? Everything is used and recycled, especially the carbon compounds. Okay, so cysteine itself, okay, is actually created from methionine, okay, and so what you do is, again, you're going to acetylmethionine, and then what you do, so with that acetyl homocysteine, right, so we're going to lose this amine here, so you go from the acetyl homocysteine, and then you hydrolyze this to the homocysteine. So now you can either do one of two things. After you can either recycle this back up to make a new methionine, or what you can do is you can react it with another serine. Okay, so so this is from the homocysteine, and then this part here is from the serine. Okay, and so literally what you're doing, and then what you need to do then is just hydrolyze on this side here. If you do that, then you get the you can get this cysteine out. Okay, so it's literally passing the sulfur from, you're connecting it in, and then so this is bringing the cysteine, this is connecting to it, and then this is the other side is the one that grabs the serine, or excuse me, the sulfur out. So. Okay, so phenylalanine, tyrosine, tryptophan. Okay, so with that, so these are actually going to use what's called chlorosmate and shikimate, with that, and so these are very complicated processes where you come in here and you hook these two sugars together, phosphoenolpyruvate and from the glycolysis, and erythrose 4-phosphate, right, from pentose phosphate pathway. If you hook those together to make a 6-carbon sugar, you can then cyclize that around, and then it's a process of trying to form this the benzene ring. Okay, so you come in here, again, another phosphoenolpyruvate is going to come in here to add this part in here to make the chlorosmate. Okay. Now, so for this, so this part here, this cycle here is actually you can actually this is obviously going to be important in plants because they have to make all their own amino acids. And so what this does is it blocks this step. some of that course mate biosynthesis. So if you block this, then you're going to be able to you're going to be able to shut down amino acid biosynthesis in the plant, and hopefully it's a weed, some of that. So in the weed, and so if you can't do that, then the cell's going to die because it can't make any more amino acids. This enzyme isn't found in mammals, right? Because these are essential amino acids, and so we don't have to worry about this blocking this enzyme. Now, it is controversial because we do use really high concentrations in the fields, and so as well as for insect populations, stuff like that. And so we won't get into the politics of all that, but it was designed to block an essential amino acid, stuff like that, something that we don't have but plants do. The so from the course mate, right? So we can come in here, we can make the prophenate, like that. So then we have two options. We're going to decarboxylate here no matter what. Okay, to make to here, to there, or to here. Okay, but the question is, which one of these do we take off? Okay, in the process, if we take off the OH, for that, then we're going to make phenylpyruvate, which then we can use to make phenylalanine. Or if instead we remove the hydrogen here, then we can make the hydroxyphenylpyruvate, which then we put in here. We add the amine to make tyrosine. So which one of these do we remove? So to make either one of those. Now for tryptophan, okay, course mate, it's a little bit more complicated because what we have to do is we're going to use the course mate, right, that we used before, but what we're actually going to use, this part here is not actually going to be part of the sort of, this is actually going to be used to make just the side chain for this. And so what you end up doing is you end up coming down, and this course mate ends up making the indole that you use for the tryptophan. So with that, and then we use serine as the amino acid part. So with that, so we sacrifice the amino acid, the serine. We lose the OH on serine, and that as well as this hydrogen here to lose water, and then you end up making the tryptophan. Tryptophan synthase, okay, so it actually has an alpha and a beta component to it, and there's actually, what you end up doing is you end up it connects the this through here, and so this part here actually creates the indole, and then down here this PLP, that's housing the serine, and so this can get transferred down to this part, and so they connect through. But because they're connected, okay, you don't want that indole to react with something else, okay? And so what ends up happening is this yellow indole synthesis, after that's made, then it actually travels down a tunnel down to the, you can see this on here, the magenta, the cofactor here. So there's a 25 angstrom pore that goes through there. And that it allows, it's complicated to make a pore like this, but what you can do is it provides, it allows the enzyme to be able to hold on to the amino acid in order to be able to, so that it doesn't release. So it's much more, so you don't have to worry about diffusing out, finding the enzyme, and then diffusing back in with that. And so it's a faster. It also allows you to react, keep a hold of, and handle reaction intermediates that may be very reactive, okay, because they're not going to get exposed to the water environment and other molecules that can cause the molecule itself to be able to get broken down. Okay, so how is this, so how are these processes controlled? There's a few different ways with that. And so the first one is through the committed step. So this is the first irreversible step. So remember this back from glycolysis. It's the first So if you go over the cliff on this one, there's no going back. You gotta do this. So if you want to, so what you want to do is you want to, that's where you want to regulate it, because you if you don't commit to the process, right, to that pathway, then you can go in and use those components for something else. And so what you can do is you can then do what's called feedback inhibition, where this product here, down the road can come back and inhibit this enzyme. If we're getting plenty of this product, we don't need any of these intermediates, so we can inhibit back here to this committed step and say, okay, we're fine. If this is if we shut this down, okay, no more B is produced, then we can do C, D, E, F, until this gets starting to get siphoned away. Then we can start producing more. Okay, you can also, if you've got one intermediate, so that makes a couple of different ones, you can actually do this. You can separate the flow. Okay, let me go back and so you can see this. Oop. And so what you can do is you can say, okay, we don't have to necessarily control here, but we can control the production of one of the components. Okay, so if we turn on this synthesis, that there's a lot of this, then what we end up doing is we end up going down this pathway here. But once this starts to build up, then we can shut this down. If there's none of this, then what we can do is, well, if there's none of this, then what we do is, well, we can just cycle it into the pyruvate and then be able to produce these amino acids here. Once we start building those up, then these can come in and activate this process again, make a whole bunch of this, and so it ends up going down here. And so by varying the concentration of this, we can actually tell whether or not, if there's a lot of it around, we can go down this process. You can go down this way. If there's a lot of this, then we can go down that way. Okay, you can also use different versions of the same enzyme, some of that. And so, so if you want to have, so if you want to produce this, right, one version is threonine sensitive, the other one is lysine sensitive. And so, again, if you build up a lot of this, then this version will be, this version will be inhibited. So only this one will be active. If this one starts to build up, so that, say, threonine builds up, this get activated, so that only this one gets produced or is able to get active. So you can still do the chemistry, but it's only going to be shuttling one way or the other. One version is going to be active at a time. The other thing is, and again, you don't need to know this chart, as you come through here, right? So all of these processes can come back and inhibit really early stage biosynthesis. So things like glycolysis and production of glutamate and that kind of stuff. So these are all sort of different feedback inhibitors. So you can use these as a precursors for a lot of different things. And so adenine and cytosine, right? DNA and RNA, histamine, NADH, epinephrine, serotonin, sphingosine, all this blue is those atoms are from amino acids. Okay. So with that, so the last thing you can use these amino acids to make molecules. So with that, so the classic one is glutathione, right? It's a tripeptide that if you have two of these can come in and make a disulfide and be able to get reduced. And so this one, glutathione peroxidase, uses a selenocysteine, and what this does is that if it encounters a toxic peroxide, it can oxidize itself, and in doing so, it'll reduce down the peroxide down to an alcohol. So if this gets oxidized, then we need to reduce this down, and so to do that, we actually use the one glutathione to make this to make the selenosulfide, and then we have another glutathione come in here, reduce this down further to get back to the selenate of that process, and in doing so, we're going to reduce down the glutathione down to the disulfide. Okay, so it's the selenomethionine that does the actual chemistry, but the glutathione itself is what is what we sacrifice in order to be able to to do this chemistry. You can also do this, do these to make porphyrins, and so the classic one is they all have this sort of general structure, some of that these aromatic structures, and the classic one is heme. And so if you take a succinyl CoA and a glycine, when you combine those two together, you can make the gamma amino valinate, right? So for that, and so it is an amino acid. It's just they're not right next to each other. Okay. So this comes in from citric acid cycle, right? Glycine from amino acid biosynthesis, and you can start stitching these things together. If you put 8 of these things, so 8 of these can come in to make 4 of these. You stitch those together, and then this comes in to sort of origami it sort of in here to make this classic sort of porphyrin structure, and then it's a process of just getting the double bonds where you need them. Okay, so the classic one is to make heme. Okay, and so when you get a bruise, right? So what's happened is that the blood vessel has ruptured and you're releasing in the red blood cells. That releases heme, and so your body's going to start breaking this down. Okay, to be able to reuse those components. But when that happens, it bursts open, it sort of bursts open that ring, the porphyrin structure to make this, and even though this is still when this is really conjugated, some of that it makes, the compounds called biliverdin and bilirubin, some of that. So as these things get released, right, you release the iron, stuff like that. So what happens is this. What is it? And so these are colored, which gives you the sort of browns and yellows and that kind of stuff when you get a bruise, stuff like that. So the last thing is if you'll sometimes see newborns put under a blue light, stuff like that. And so this one is, you naturally want to recycle the heme within within the liver. If the baby's liver isn't quite up and running yet when it's born, you'll have this buildup of bilirubin, some of that, and so what they notice is that if you put it out in sunlight, some of that, it'll break it down. It basically helps the newborn's liver get up and running, some of that, and so which will come in you'll have the newborn will come out slightly yellowish, so jaundiced, because the liver isn't running. So they put them under instead of taking them outside with patches, things like that. You can have it in a more controlled environment. You put them under the bilirubin, this blue light, for a little while. That light is able to actually break down this to more reasonable levels in order to be able to help out the newborn. Okay. Thanks, everybody.